Welcome to the Bodhi Academy. My name is Shrutika and today let us continue with our part 3 of our chapter Sexual Reproduction in Flowering Plants. So today what we are going to study is the female reproductive part, that is the pistil. So pistil is the female reproductive part and pistil forms what? The female gamete, that is ovules. And how these ovules are formed? It is formed through the process of megasporogenesis. So today we are going to learn about the pistil, that is the female reproductive structure, then how the megasporogenesis process happens to form the gametes, and how we will study this in detail now. So first let us see what is pistil. So the pistil, the gynoecium may consist of single pistil, that is monocarpellary, or more than one pistil, that is polycarpellary, which may be fused, that is syncarpous, or free, that is apocarpous. So pistil, we all know that pistil consists of three parts, that is stigma, style and the ovary. If the pistil is it consists of only one single pistil, then it is called as monocarpellary. If the pistil has or more than one pistil, if the flower has more than one pistil, then it is called as polycarpellary. If the pistils are fused, then it is called as syncarpous, and if the pistils are free, then it is called as apocarpous. So each pistil will have three parts, that is the stigma, style and the ovary part. Ovary will consist of the ovules, that is the female gamete of the flower. So this is how the structure of the pistil is. So now let us see how the structure of ovule is, that is how the structure, what is the structure, which are the different parts of the female gamete, that is the ovule. So ovule, which is called as the megasporangium. So let us see the structure. The ovule is a small structure attached to the placenta. The funicle, the first part of the ovule is the funicle, which is a stalk by which ovule is attached to the placenta. Then comes the hilum, which is a junction between the ovule and the funicle. Then integuments are the protective envelopes. Then micropyle, a small opening at the tip of the ovule into where the pollen tube enters. So chalaza, the basal part of the ovule, and nucellus, that is the mass of cell enclosed in integument, has abundant food reserve. So these are the different layers of the ovule and their functions, as in how this layers functions in the ovule. Now let us see the structure of the ovule. So this is the structure of ovule. So this is the ovule structure. This part over here is the funicle, which helps in attachment of the ovule to the placenta. Then this is the hilum part, micropyle, which is opening wherein the pollen tube which germinates will enter into the embryo sac. So this is the embryo sac. Okay, these are the integuments which are helping in the protection of the ovule. This is the chalazial pole, wherein this is the basal part of the ovule and the funicle. And this is what? The nutritive layer, which consists of abundant food reserve inside the ovule, which helps in what? Which helps in the providing nutrition to the growing embryo inside the ovule. So these are the various parts of the ovule and their function. Now let us see the process of megasporogenesis. So what is the process of megasporogenesis? Megasporogenesis is the process of formation of megaspore from megaspore mother cell by meiotic division, and this process is known as megasporogenesis. This process takes place in the ovule. So inside the ovule, this megasporogenesis process takes place, wherein the formation of megaspores takes place inside the ovule, and this process is called as megasporogenesis. Then how this process happens in the ovule? The ovule differentiates a single megaspore mother cell in the micropylar region of the nucellus. The megaspore mother cell undergoes meiotic division that results in the production of 4 megaspores. In most of the flower plants, the megaspore develops into a female gametophyte, that is the embryo sac. The nucellus of functional megaspore divides meiotically to form two nuclei which moves to the opposite pole to form two nucleated embryo sac. Two more sequential meiotic division results in 8 nucleus, and 6 of the 8 nucleus surrounds around a wall called as the egg aperture. 3 cells are grouped at the micropylar end to constitute egg aperture, and 3 cells at the chalazal end to form the antipodal cell. At maturity, embryo sac is 8 nucleated and 7 cells. So let us see the megasporogenesis process in the embryo sac in detail now. So this is how the process of megasporogenesis happens in the flower. That is, this is the nucellus tissue. Okay, one nucellus tissue will divide to form what? The megaspore mother cell. This megaspore mother cell which is there, it will further divide to form megaspore dyad, that is 2 cell. Then this will further divide to form a tetrad. Okay, from this tetrad only 1 will survive and 3 will be degenerated. Okay, this will further undergo karyokinesis, that is nuclear division. Now the cellular division has happened. Now what will happen is the nuclear division will take place. That is, this single nucleus which is there, this megaspore which is there, which is formed from the megaspore mother cell, this will undergo what? The nuclear division. Okay, the nuclear division will result in formation of 2 cells. Then these 2 nucleus will further divide to form 4, and then it will divide to form what? 8. So from 2 to 4 to 8. So this will here at this stage the nuclear division will take place. Then what will happen is the three cells from this part, 3 cells will move at this point. Okay, and 3 cells will move at this point, and 2 will remain in the center. Okay, so then what will happen is wall formation will take place. That is, wall formation around this 3 nucleus will take place, and what you will get is a mature embryo sac with the following compartment. That is, 3 nucleus will form a cell, 3 layers, 3 compartment like thing, and this will form at the chalazal end, and these 3 cells are called as the antipodal cell. Then at the micropyle end, what is formed is the synergid, that is the 3 cells which have come down at the micropylar end. 2 cells will form what? The synergid, and 1 cell will form the egg aperture, and the 2 cells which are there in the center will form the polar nucleus at the center. And this is called as the mature embryo sac, which will undergo fertilization and will form the fruit and then further seed. This is how the megasporogenesis process happens in the flower. So what we have seen today is the structure of the ovule. We have seen what is the pistil, what are the types of pistil, and the process of ovule formation, that is megasporogenesis. So thank you for watching today's video. Please don't forget to like, comment, share and subscribe to the Bodhi Academy.